He's an associate professor, reader at Imperial College. He did his PhD at TU Munich. He did his postdoc at MSR. He's really, my mind, spearheaded a lot of things in machine learning for medical imaging. I remember reading a lot of his work on decision forests and discrete methods for image registration, which actually kind of got me into registration back before deep learning was big. But then in deep learning, he's done a lot of really important work that's still widely popular and cited. Like DeepMedic was one of the first popular architectures in our field, still widely used for deep brain lesion segmentation and so on. And he usually does very creative work. So today he's going to talk about some super interesting sort of first steps in causality and uncertainty, probably based on the title. So Ben, I'll let you take it away.
Yeah, great. Thank you very much, Adrian, for the introduction. So apologies for those people who have attended the Cornell talk. The first part will be very similar. I will go through it a little bit quicker so that we get to the latest and recent stuff and can talk about this. But there are also things along the line that I hadn't time to talk about in more detail, which relates to this last part, the generalization part. Let me see if I can have a laser pointer here. Okay. All right. So just some disclosures. I have to put them when I speak at medical conferences, I guess, these days. But this has nothing to do with this talk, actually. So for those people who haven't been to Imperial, so this is Imperial College campus, the main campus sitting in the heart of London, central London, surrounded by nice buildings. So there's the Royal Albert Hall and the National History Museum. And the Science Museum, I think for maybe some people who have attended MICAI 2009, I believe it was here in the Science Museum. So this is our Queen's Tower room. Yeah, so there are nice buildings around. There's the Hyde Park, and we are here, Department of Computing, which is one of the ugliest buildings in the area. Anyhow, so this is the Biomedia Group, a large group founded by Daniel Rückert many years ago. And there's a team within there that I'm leading, and everyone in this group is kind of now working at this intersection of medical imaging and machine learning, I guess. So these are how I would currently describe my research themes, if I have to put categories on them. So there's some work on core algorithms for what we call semantic imaging, but let's say things like image segmentation and so on. So this is an example of this DeepMedic architecture. We do some work on image-based detection of disease, and more, I guess, machine learning methods, really. So I have quite a few people who are actually now more on the core ML side and want to do more a bit on theory and methodology and so on. Also some work on robust and reliable machine learning and so on. So for DeepMedic, there's a recent demo that we put out. So if you go on this website and want to do some live CNN-based image segmentation of brain lesions, that's where you can try it. So my first part of the talk is about the causality things that we started to talk about, I guess, over the last year or so, and really also just started to think about it. So it's a really recent and a new area. So I'm not coming from that field. I'm not an expert in causality, but we got interested in it and wanted to understand how it relates to medical imaging, or to the problems in medical imaging. And I talk a little bit about the general problem on generalization. So this starts basically by looking at what probably most of maybe you, or at least in our group, people are doing, which is predictive modeling. So we often try to work out models that can be trained to make predictions. And in our case, it's typically the input is an image. So we have an image X going into a trained model that then can make predictions related to some annotation Y. So we are typically interested in learning this conditional distribution, right? So given an image, what is the probability of a certain annotation? And the assumptions we make here is that we have sufficient training data, so pairs of X and Y available. And the big assumption that we do make is that the training and test data do come from the same distribution. Otherwise, the model is not working well. It's not robust to changes in these data distributions, or at least not easily robust unless we do stuff. So there are some challenges when you try to build these models. So in particular in medical imaging, we often deal with quite small amounts of training data. And I know Adrian and others are very interested in how we can make or how we can train or learn models just from a very limited amount of data, maybe even just a single image. But we generally have this problem of data scarcity. So we might have this data distribution P of X, but we only have a very few points for which we have high quality annotations. The other problem is that we have data mismatch. So this relates to this assumption that training, so the distribution over our training images and the distribution over our test images come from the same distribution, which is often violated in practice. So you can have things like population shift, right? So your patient population might change. The disease prevalence or the label prevalence might change, right? So you have this label shift maybe. The other big one, I think that we are actually not talking a lot about, or maybe not enough, at least in our field, I have the impression, is the problem of sample selection. I know in neuroscience, they talk about this much more, I think. But generally, when you do machine learning, you rely on a training data set, and you have to really think carefully about how this training data set was actually generated and what the data collection was like. Because typically what you have is that your training data is only a small subspace of maybe the entire distribution on which you might want to test your model later, or where you want to deploy your model. So these are all challenges I think we all face, and there's lots of work both on the ML side, but also if you go to MICAI or Middle, you will see lots of papers talking about tackling some of those challenges or all of them. So what we tried to do, I guess a year or maybe more than a year ago, when we got interested in causality, is actually can causality give us a new perspective on those challenges? So that's what we tried. So here, a very brief background on causal reasoning. Very simple scenario. We have a cause A and an effect B. And this is already a causal diagram, right? So if you draw this directed arrow here, it means that there is a mechanism that goes from A to B. So if you have a cause, there will be an effect, and this is what we call a mechanism. And there's a big assumption. So there's this principle of independence of cause and mechanism, and we'll go back to this, which basically states that if you know about your distribution of your causes, this is really independent of the mechanism. So the mechanism alone exists. You can think of these maybe in a healthcare setting, like if we have some symptoms on the left side and maybe a diagnostic test on the right-hand side. So if you go to your doctor and say, these are my symptoms, I have a cough, I have chest pain, they might give you a certain, or they might indicate a certain diagnostic test, let's say a chest CT scan. And this is typically done based on guidelines. So they often sometimes really just look them up. So there are these decision trees that say, okay, these symptoms mean this diagnostic test. And you can just think about the distribution over the symptoms doesn't really change this, right? So another example is in physics. Laws of physics are mechanisms. And let's say this is altitude and temperature. It doesn't really matter whether you're in Switzerland or in Boston or in London. The mechanism going from altitude to temperatures is independent of like the distribution over the altitudes. So going back to imaging, so we wanted to know is what is the relationship between images and annotations? And interestingly, we couldn't find much literature on this. There's a little bit of literature in the computer vision domain where people talk about causal relationships between annotations and images, but in medical imaging we hadn't seen much. So and interestingly, there are two scenarios. So either you have a causal relationship, right? So the image causes the label, and that means you're predicting the effect from the cause, right? Because we are still interested in this conditional distribution. Or there can be an anti-causal relationship. So that would mean you predict the cause from the effect, right? So we're changing the relationship here. And we wanted to know if those actually exist in our world of medical imaging. So we looked at some examples. So we discussed some, we came up with these examples. Skin lesion classification, we would argue that we have an anti-causal relationship. Because you have an image, let's say a demoscopic image, and maybe your ground truth or your label is as close as it can get to ground truth because it's biopsy-derived diagnosis. So this would really be like a good proxy for the true state of disease, right? And you could argue that the disease causes the image, right? So if you look at those images, they look like because you either have a benign or malignant cancer. So you do try to predict now from image to disease, so basically in reverse to the causal relationship. Then we argued that in maybe other scenarios, like brain tumor segmentation or general segmentation problems, maybe it's the other way. Because what we are trying to basically to do when we learn the mechanism going from image to annotations is just to mimic the annotation process. And here we would argue that actually the image is causing the contour because someone is sitting down with the image and drawing a contour. So while the disease is still causing the image is causing the segmentation that we're trying to predict. So the prediction goes along the causal direction. Then we argued that there are some cases where it's really difficult to make up your mind whether it's causal or anti-causal. So an example is chest X-ray, and you have your diagnostic labels extracted from a report. Let's say even using maybe something like natural language processing. That's a little bit tricky because you don't really know where that diagnosis is coming from. It could be derived from the image. Then you could argue it's a causal direction. But maybe the label is actually coming from another blood test, a lab test, something that's even more important than the image. Maybe then it's the other way around. So what we argue is that you need meta information. You need information about the data generating process. You need to know exactly where those labels come from in order to establish the causal relationship. Now, good. Let's say I convinced you that there are these two cases, causal and anti-causal, in our world. Does it actually matter? And going back to the challenges, we argue that it does. So for instance, in data scarcity, where you have labeled data is scarce, unlabeled data is abundant, people use things like semi-supervised learning, right? So it's a hot topic. There are lots of papers. I'm sure there will be lots of Mikai papers on semi-supervised learning. Actually, interestingly, if you look at a paper by Bernhard Schulkopf, who discussed recently, but also over the last years, the idea of causal and anti-causal machine learning is that from a causal perspective, there's actually a difference. Because you're in all cases interested in this conditional distribution. But if you're in the causal setting where you predict from X going to Y, what it means is you actually have your unlabeled data. So the images for which you don't have segmentations are the causes. And you're trying to learn these mechanisms. And remember, there's this principle of independence of causal mechanism, which just taking those statements should maybe mean that semi-supervised learning shouldn't work in image segmentation. Or at least that just like taking the naive approach of the typical SSL approach is you try to fit the decision boundary into areas of low density where there's no data. Maybe that doesn't work. So that's, I put a question mark there because I think this is open for discussion and people have controversial views on this one. But we thought it's an interesting thing, maybe some food for thought. On that line, you can also argue that data augmentation always works in both causal and anti-causal settings because you're basically trying to generate these realistic pairs, right? So you're basically trying to get information about the joint distribution. And that should help in both the causal and the anti-causal setting. And people are even going into this direction of learning how to augment. There are some examples here. I know Adrian is also looking into this, which I think is an interesting direction. So the unlabeled data here gives you the means for learning what realistic perturbations you can make. So maybe that's more promising than doing SSL in image segmentation. All right, so I quickly go over dataset shift. Just from a causal perspective, the only thing that we want to say here is that causality allows you to categorize better what type of shifts you might expect. So what we try to do in this work, where we argue that causality is an interesting thing to look at for medical imaging, is that you suddenly can use causal diagrams to clearly communicate what type of shift there might be. So here we are doing this by introducing another variable Z, or Z, which is the unobserved true anatomy. So it's the physical manifestation of the subject that you want to scan. And we use a indicator variable, a domain indicator D here, to say which of the variables is affected. Now remember, we can either be in the causal setting, so we have image X, label Y, we're always predicting in this direction. Now, depending where we put this domain indicator, we get different types of shifts. Or this is one way of formalizing different types of shifts, let's say. So there can be population shift, right? So the true anatomy might change depending where you are. So where you are means you could be at train or test time, you could be at development or deployment time, but you might have population shifts. So the anatomy that you're seeing, the prevalence might change, right? So you might have this, let's say, younger or older cohorts at train or test time, for instance, and that changes your image. Interestingly, this is not a big problem because the mechanism itself that you want to learn, so going from X to Y, let's say, to do image segmentation, should be valid still, even if your population changes, assuming that you actually have training data that covers, I guess, the support of the distribution that you want to cover at test time. If you don't, then it's difficult. Acquisition shift means the image is affected, depending where you are. So this could be you have an MRI scanner for training, and later you want to test on GE scans. So acquisition shift is a big one, I think, in our community. Annotation shift is a terrible one. It basically means that between test and train, the procedures for generating the labels has changed, and there's little hope that you can actually learn in these settings, right? So that's why it's important if you do multi-center studies and so on, that you have good standardized operating procedures and so on, that people follow the same ways. I mean, just imagine someone labels something with a different category consistently between two different sites. There's no way you can do classification in that setting. In the anti-causal settings, it's similar. So I will skip them and you can have a look. There's, of course, also acquisition shift and some others. All right. So the different shifts have different problems and also different solutions. So that's why it's important to find out in your task that you're trying to solve what type of shifts you might expect. And then you might also want to consider possible mitigation strategies for that. I just want to go back to this brain tumor segmentation. Why is causality cool? Because if you actually draw out the causal diagram that you assume is there, you can also then communicate that there might be an issue. So if you draw in the domain indicator here, you could say that I might expect that between my train and test domains, I get a different population, right? So this would be population shift, this would be acquisition shift. So the whole thing about these causal diagrams is at this stage is about communicating your assumptions. So I quickly want to mention some techniques that we looked at over the last years to mitigate, for instance, things like acquisition shift. So this is a technique that Kostas, one of my first PhD students, was working on. So this was, I think, at IPME 2017. Where we looked at adversarial training to make a segmentation network agnostic to the domain. So the idea is the practical scenario we had was image segmentation of brain lesions and MRI, and the imaging changed between what we were training on and what we wanted to test on. So it's a technique that we borrowed. So people proposed this for domain adaptation to use a domain discriminator. They hadn't done for segmentation, but that's what we did here. And it's basically you train your standard segmentation network. So this is a CNN or DeepMedic that does image segmentation, but at the same time you have a domain discriminator that can look at different feature maps at different levels of the CNN, and we evaluated which levels are the best. But basically it's just saying that these features that are extracted at training time should not allow you to tell where the data is coming from. So what it requires you is that you don't need labels for the test domain, so you can use unlabeled data from your test domain. That's great. However, you need that data at training time, right? So you train on two domains simultaneously. One of them is labeled, one is unlabeled. The unlabeled goes to this loss together with the labeled ones. The labeled is only used for the segmentation loss. And that works surprisingly well. So we tested this on a quite severe scenario of domain shift, where at the source domain, they used the multi-sequence MRI. So there's a gradient echo scan here, which shows microbleeds. And then later in the newer protocol for this application of traumatic brain injury, they switched to something called SWI, which is a different MRR protocol, but clinically it's the same because it also shows microbleeds. But for anything that you learn, it's terrible because it has different contrasts. Like it shows it slightly different, and that usually means that your trained network on gradient echo would fail if you suddenly give it an SWI scan. So you can see this if you train just naively, it basically fails to segment any of those lesions. These are the manual ones. If you just say, okay, there's one of the modalities is not matching, right? I still have flare, I still have T2 and so on. Maybe I can just discard it. Yes, you can, and then you get better, but you miss the microbleeds. And here, if you do domain adaptation, trained on gradient echo, trained on SWI, you still are able to detect those microbleeds. So that was quite nice. I'm skipping the die scores, but you can have a look later at this. So as I said, this requires you to have things at training time ready. So you need the second domain at training time. So the next step would be to look into what we call domain generalization, or what people call domain generalization. So here the idea is, how can you generalize to new domains that you haven't seen, right? Where you don't have training data. So you will only see it at test time. So this has other assumptions. You need more than one domain at training time. However, you don't need the domain where you later will test on. And it's based on a very simple idea. You basically simulate domain shift during training by using the data from, let's say, the 2 or more domains that you have available at training time. So you try to make your network more robust to domain shift by simulating domain shift during training. We tested this mostly on computer vision data. Initially, there's a benchmark for domain generalization if people are interested in this. One is called PAX because it uses photo, art, cartoon, and sketch. So it's a quite severe domain shift. It's basically the same categories, same 7 categories of objects, but across very different types of images. So photo, art, cartoon, sketch. So you can see the matching things here, but they're quite different. So what we looked at is, so we called it learning of semantic features, but basically what it tries to do, or what it combines, are several things. So it's a so-called episodic training procedure. We'll talk about what that is, and a new loss function. So the first thing that we have to do in order to look at this, how this works, is so we take our model, a CNN, that does classification, and we extract basically, or we decompose it into what we call the feature extractor and the task network. So it's basically just your CNN. At some point you say, okay, up to here, that's my feature extractor, and then you have the last bit that does the classification, right? So their interface is this representation set. By the way, I should say, if there's anything unclear, just interrupt me. I hope people are still following. It's interesting if you don't have any feedback. Okay, so
Wait,
we have these. Yes.
Can I just interrupt? So we had
sure.
some questions earlier.
Sorry.
Earlier on the causality, but I wasn't sure at what point. Like maybe I thought I would wait till you end the causality.
Yeah, any please.
Okay. Well, Raz, do you want to ask your question? If you're.
Yeah, sure. So I was wondering, do you have to pre-specify the causal direction beforehand? Sometimes it could be unclear exactly if it's causal or anti-causal. I was wondering if you can even learn it, if you can learn the direction somehow. So, and if it's feasible in practice to do this learning.
That's a good question. So this is about causal discovery. It's a whole research area that people look into, and it's difficult if you do it from observational data. I think with certain assumptions you can do it. It's not something we looked at all. So what we are saying is this is so far it's all about making your assumptions visible. So you have to decide. And that's where domain knowledge comes in. Now, this is why causality is also a big topic of debate. And there are people who say, no, we should not do this. We should just look at the data and correlations, right? Because here we are trying to go beyond correlations. And the observational data doesn't tell you the causal direction. You cannot tell apart, right? But often we do know. I mean, for instance, in the case of skin lesions, I think it's relatively clear that the disease causes the image. So if you're in domains where you have knowledge about this, the question is, why not using it?
Yeah, thank you.
I just had like, maybe this is a very silly question, but I'm thinking, you know, if like a student wants to approach a segmentation problem, let's say, or something like that, is it like, I think you went over how causality, you know, these causal diagrams can help us think about the problem, but I'm wondering if do you feel like it helps like it changes how people might approach, let's say, a segmentation problem by having thought about the causal relationships? Like, is like in the process of doing research, is that?
I hope so. So I will invite you later. I think it starts to help you when you start thinking about I would just call it the data generating process. Simply because, I mean, there are works like, so I think Lena Meyer-Hein did this work a couple of years ago where they looked at biomedical challenges and the problems and challenges and why can we not always trust the rankings and the best performing algorithms and so on. And we sometimes lost touch with like just looking at the challenges and chasing dice scores and so on. And the problem they discussed was there was so much information missing about how the data was generated and so on that you can't conclude who's the best or which one is the best algorithm. So just by sitting down and thinking about your problem, how the whole data comes together, and I think the big promise in that is that you will identify problems. So you will identify these things. Oh wait, there could be a data set shift. I have to tackle that, right? Otherwise, my thing will never work in practice. So hopefully yes, but yeah. I don't have a good use case yet where we used it and that made a clear difference.
Sure. I mean, it's a bit of a soft thing to measure, right? It's hard to measure how much difference it made. But yeah, okay. Thank you. I don't think there's any questions right now. If anyone has questions, please either interrupt if Ben's okay with that, or put them in the chat.
Yeah, just show. I don't see the chat at the moment, but
okay. Okay. Thank you.
All right. So quickly going through this. So episodic training. What it means is that so I said you have more than one domain, right, at training time, and what you do is you split them into what we call meta train and meta test. So if you just have two, you basically alternate. Sometimes one of them is the train, one of them is the test, and it just means how you update. So these are the parameters of the networks, right? So this is the feature extractor, this is the task network. So together they are all the weights of the CNN. And if you do your typical gradient descent and you update your networks based on the task loss, right, the classification loss, we are saying that you update your network, so you get new parameters prime, and then you use this updated network later on to evaluate the losses that I will talk in a second, instead of directly updating your network and evaluating on the train data, right? But you evaluate on the meta test data. So you use a different domain for computing the gradient, and then the other set to compute the losses in the next round. So that's what's called episodic training. So this was proposed in this paper here at the bottom. Now about the losses. So I will basically just try to explain them on the figure here. So let's ignore the math for a moment. But basically what we have, the feature extractor, right? So we have two domains at training time. And the idea is for this first loss to preserve interclass relationships. So we call it the global class alignment. So basically you stay within a domain on both for the two sides here. So this domain gives you features, this domain gives you features. And then you compute the interclass relationships after you pass it through the task network. So you get kind of affinity matrices between the classes. And what you say that those affinity matrices should be similar between the two domains. So basically it just means intuitively things like if you misclassify on one domain a dog and a cat with a certain probability, and then a dog and a house with another probability, you want to preserve this, right? So in the other domain, dogs and cats are probably also closer than dogs and houses. That's the whole thing. The second loss, or the second part of the loss, is a local sample clustering, which is very related to actually semi-supervised learning, if you want, or a recent work on contrastive learning. And it's actually, we did use contrastive loss as one option, or a triplet loss. And here the idea is that you now say samples that are from the same class but different domains should still locally cluster together. And for that, you use a small extra network that basically just computes a distance between them, an Euclidean distance after you passed it through that network. So it's a metric learning approach. So it basically says you see these different shapes mean they come from different domains, but the colors are the classes. You say that, samples from the same class should go into the same area of the feature space, even if they're from different domains. So interestingly, so this approach that we came up with all these three things: episodic training, global class alignment, and local sample clustering, improved over state of the art on this benchmark. So that was interesting. But also, I think an interesting take home was we did a baseline where we just did a CNN and training on, like, naively all the available training domains. And if you look at the average performance, it was better than some of the previously proposed domain generalization methods, which I guess is a nice point on sometimes you need to carefully look at what baseline you use to compare. Okay, so we did an ablation study. We wanted to know how these different, whether it's episodic training, global loss, or the local loss. And it actually turns out that you need all of them to get the best performance. That was quite nice to see, that they are really complementary and helping with that. But I guess the numbers otherwise don't mean much here, but just says that it kind of worked. Except that, so when we also tested it on a medical imaging example, tissue segmentation and brain MRI, it didn't make a massive difference. So we had 4 different MRIs, so MRIs from 4 different sites here. So the typical thing, if you compare, so training on a single site always gives you the best DICE scores. If you train on this and test on the other, you see how the DICE scores drop and so on, right? The thing is that if you train this deep all, so basically this means you train on 3 of the domains and test on the whole load domain, you're already quite good. So our domain generalization improved a little bit on top of that, sometimes okay-ish with like 0.7 DICE or something, or 0.5 DICE, but it's not massive, right? But I guess that's because the performance is already quite good.
Ben, can I quick interruption? So Maggie, do you wanna ask your question? Or
sure. A quick question is, we're still, like, when we're looking at the feature extractors and like we're making sure that the interclass sort of like errors match, we're still looking at data from 2 domains. Is that correct?
Yes.
So why, like, where is the part that allows us to generalize to new domains? Because if those 2 domains that we're using to train are roughly sort of like similar or close together, then we're not really spanning the entire sort of support of possible images. Is that sense?
That's a very fair summary. So, I mean, yes, your domains that on which you train will heavily define how well you can generalize afterwards. To be honest, I think when we talk about generalization, I think it's often not about extrapolating, but you basically hopefully can better interpolate between the like areas that you map out, right? So if you think about the representation space, one domain is here, one domain is here. There's very little hope that afterwards you can do well on a domain that sits up here somewhere. If you can see that, I don't know.
Yeah, that makes sense.
Yeah, I guess, yeah.
So for medical imaging, you didn't. So these were all the same modality, just different sites. So T1.
Yeah,
something,
all
or?
T1 weighted, different sites. I think one of them, so the one that performs the worst is, I think, a different Tesla, I believe. I have to double check where they
okay, so it's all variation sort of within this similar protocol. Right?
Yeah.
Yeah. Okay.
But yeah, I mean, it's a good point. It would be interesting to test it on something where you have more domain shift. Because I think this method of domain generalization, I mean, it was working really well on this, which I find quite exciting. I mean, that you can learn on photos and predict on sketches, right? It's crazy that it works with 70% accuracy, to be honest. But even the baselines did like 65 or almost 70%. I think it's interesting that it works in the first place.
Sure.
But that's a massive domain shift. So maybe this method is really for things that are like far apart and don't matter too much if things already are relatively close. So the sample selection I was alluding to is, I think, quite important, but I don't have too much to say on it. But just that, again, causality helps you to communicate. So again, in a causal diagram, and here I'm ignoring the causal direction between X and Y because it doesn't matter for this selection diagram, but it just shows that you can illustrate in the diagram what type of selection has gone into, let's say, the training process or between training and testing and so on. And it can be random, right? There's like a randomized controlled trial, which is great, no biases. It can be image dependent. There could be a quality control on the images. Maybe not every image made it into the training set because there was some corruption, some noise, some stuff. You could make it target dependent, right? So this is often what we actually do in machine learning, where you, let's say you train on a 50-50 balanced data set. You do it actually target dependent sampling of training data. You might do it even during the training in the epochs. You might select specific like ratios of positives and negatives or something, or it could be done both on both. And this causes different biases, right? And it also has different mitigation solutions. I mean, there are ways of, like, for instance, weighting your loss functions, reweighting certain things, and so on. So there are mitigation strategies for that. But again, it's just causal diagrams, great for communicating these things. For instance, in the skin lesion classification, if you think about these biopsy-derived diagnoses, right? So you say this is close to ground truth, and maybe that's great for training. But if you think about it, so when do you get a biopsy? So in a causal diagram, you could now say, so the image, there might have been a suspicion, right? The doctor said, I'm not quite sure about this thing. So let's get a biopsy. And these are the only cases where you then actually get biopsy results. So there was already a suspicion. So maybe it's a completely biased training data set. And you have to be careful. So I guess that is the note to the PhD student who starts on a project. And sometimes, what do? We download data from like a challenge data set or some public like available data. And maybe we never thought about this, but the training data set might have like be heavily biased because of these things. And then we train, and this will never work in a screening setting, right? Because in a screening setting, you will see lots of images that were not suspicious at all. So there is a little bit of work. So if people are interested so we did a little bit of an analysis how class imbalance affects neural nets. So that was Mikhail last year by Seju, and Loic is looking into how you can correct at training, during training, with a loss function, any sampling bias is on the, if you have prevalence shift. So if people are interested, I can forward those works. So I guess, so this concludes the first half of causality. We argued that it's also important to draw these diagrams. They can help you to communicate, and there's a lot of calls about like how we should look at machine learning in healthcare and so on, and how can we do transparent reporting, how can we make things more clear what's going in terms of biases and so on. So it might help with that. We invite people to try it. We came up with a scaffold diagram that tries to capture lots of different things that you might encounter in medical imaging. Certainly there's stuff missing, and or you might want to add more nodes or different errors and so on. So I think it's a good exercise. So Daniel and Ian were leading on that one. So this paper it's on arXiv if you want to have a look. So now I want to go to the more interesting stuff. I have still 15 minutes, I guess, or less if we want questions. So that after we did this work, we wanted to see, so how can we now actually make use of this? So far this was all about communicating assumptions. And I talked about this idea of mapping out the data generating process. But if we do have knowledge, like the question earlier about what if we know the causal direction, can we actually make use of it? And that's what we try to do. And there's a theme, not that we came up with this, but people call it causal representation learning. There's a nice paper by Bernhard Schulkopf about, it's like an opinion paper, I would say, or perspective on causality and machine learning. And it's quite interesting to think about how to use it. So we tried this. So this is very recent work from basically 2 weeks ago. But we were working on this for a year, I think, until we got where we are. So the idea is, let's say you have these two subjects here, two brain scans, same age, 68 years old, subject A, subject B. Now, if I go 2 years later and I have a follow-up scan, I hope you can see this. So I'm going forth and back here. On the left side, you see quite some changes to the ventricles. You might argue there's maybe even some atrophy that you can see. I'm not sure how much you can. But on the right-hand side, not much is happening. Right? So the question is, why are they different? And I think this is coming down to like some of the core questions we maybe should be asking more in medical imaging, and I'm sure in neuroscience and other fields they do, but I don't see often these kind of questions being asked, for instance, at Mikhail and so on. But so what makes these two subjects different? I don't have an answer yet, but if you want to answer a question like why are they different, it's actually a causality question. And you need to do some causal modeling. So we did something very simple, not to answer that question, but to get a first step towards that. So we said, let's think about a problem where we can come up with a, let's say, simplified, maybe hopefully plausible causal model for brain imaging. And it's very simple because it has only these few variables, age, sex, ventricle volume, and brain volume. And they are together forming the data generating process for the underlying image. Okay, so let's assume this is half correct. Then what can we do with this? We can actually build a structural causal model. So that's what all like causality is about, building models around like the causal diagram. And we did it in a deep way, meaning that we use deep learning components to model parts of this structural causal model. So what is a structural causal model? It's a collection of structural assignments, right? So these are these Fs. So you have one per node in the graph. And you have noise variables, which are basically exogenous noise variables. So they are living outside the model. And there's typically assumed to be a distribution over them. And they are making basically the values of those nodes. So this is basically called a mechanism, right? So one of those structural assignments takes a noise variable plus the causes. So these are the parents of that variable. So age, for instance, in this case, doesn't have parents. It's just based on this noise variable. And that defines the value of that variable. Okay. And there's a joint distribution over these noise variables. And they factorize like this because we assume that all these noise variables are independent. Yeah. So for the structural causal model, there is then a unique joint observational distribution, which basically means if you have an observation X for this, so fully observed, each of those nodes has a value now. You observe something in the real world. Then this distribution for the structural causal model, again, factorizes into these terms here, which each of them is fully determined by its mechanism and a noise variable. Okay, so this is why you have this directed acyclic graph. That gives you basically this, I think it's called the causal Markov property. All right, so we have a distribution over this model, right? So what I'm now not telling you exactly, because I don't have time, is how we learn this distribution, but you can, and that's what this paper is about by Nick and Daniel, deep structural causal models for tractable counterfactual inference. So we have a learning mechanism, we have a way of learning these mechanisms for each of those nodes, and we use a mix of deep learning components, normalizing flows for all the non-imaging variables, except of sex. And for the image, we use a conditional variational autoencoder. So here I'm just showing you, so we had some true data. So this is from UK Biobank. These are, I think in this study, we had 15,000 brain scans. And we looked at these things like brain volume, age, sex, and ventricle volume, which I'm not showing in these graphs. But what you can show, this is the true data, if you plot it, at least for these three variables. And this is what our model learned. And it's pretty close. So it learned these relationships between these variables in a really faithful way. Good. So now we have just learned what the observational data looks like. But the nice thing is, and this is different to, let's say, other probabilistic models, is that with the causal model, you can now do counterfactual. So you can produce data points that are basically these answering these what-if questions. So you take your structural causal model with all your learned parameters for the mechanisms, and then you follow three steps. The first one is an abduction step. So if you now have an image, so someone gives you a brain scan of a person, and it's fully observed. So you know the age, the sex, the ventricle volume, and the brain volume. We can talk about how we get them, but in this assumption, it only works if everything is observed. Then based on this observation, we predict the state of the world. So basically what it means, we need to predict the noise variables, the values for the noise. So that's the abduction step. So that you can do. We can do this because we use invertible mechanisms. So we use normalizing flows that allow us to invert. We do an approximation for the variational autoencoder, but everything else is invertible. So we get the noise variables. Good. Then we can do action, so we can actually perform an intervention. So we can change a variable, right? So that would be, for instance, okay, this person was 48 years old. Let's put the age to 60, right? So that's an intervention. And that results in a new structural model where just that one variable has this do operator, so that's this assignment, that's hard assignment. You put a value on that variable, and that gives you this new structural model. So only the structural assignments have changed, and you use the distribution of the noise that you inferred in the first step. And now you have this new structural model, and you can make a prediction. So now you can compute the quantity that you want, let's say the image, right? So you change the age, and now you compute a new image. And that is actually working to some degree, at least. And I think this is where I got really excited when we saw this. So here's an input image. So this is sex female, age 49, brain volume, ventricle volume measured. Now, I should say, probably someone will ask, how did we get these brain volumes? We did get them from segmentations, which you could argue they are image derived. But in our scenario, we used them from 3D volumes. And we assume they are close enough to the true anatomical measurement. So it's a bit of an assumption here. And the experiments are done just on 2D axial slices. So they are actually, you cannot measure this brain volume in that 2D slice. So, but yeah, bear with us. So you can do interventions. So if you change the sex, there's a slight change, not too much. So it's just a little bit of change on the overall brain volume here. If we change, so this was 49 years old, now we go down to 40, not much changes. We are relatively close. If we make that person older, we see what we kind of expect. So the brain shrinks and the ventricles grow. Right? So this is what you see in the difference image. So larger ventricles, overall a little bit smaller brain volume. If you just change the brain volume, everything else stays constant. You see this nice effect that, yeah, the brain volume goes down, but the ventricles didn't change. Or at least not, sorry, the ventricles also go down, right? So because brain volume has an effect on ventricle volume, right? So everything goes down a little bit. So it's nice that you see a difference between doing age and doing brain volume. If you do, so this was smaller brain volume, this is larger brain volume, so you see the opposite, which is good confirmation. You can also intervene directly on the ventricles, and then you see you can make smaller or larger ventricles. And you see this is now suddenly much more localized, right, than the previous ones. So changing age has a global effect on brain volume, right? Exactly like what you see in the structure causal diagram. So this I think is quite cool. In the paper we also did some other examples with MNIST and so on. But if you want, you can try this. If you go on the GitHub page, there is a link to a Google Colab Jupyter notebook where you can run exactly this with a couple of brain examples, and you can change yourself the variables and so on. All right, if you're interested in more on causality, there is a Mikai tutorial together with many colleagues where we want to talk a little bit more about how, or discuss how this might be a new, nice, I guess, direction to go into. So now I want to check with Adrian because I have a part 2, which is probably 5 or 6 more slides, so it's a much smaller part. But I don't know how we are on time. I guess we are.
I think we should. I think you should do it. I mean, I think if people need to trickle out, that's fine. So I don't see harm. Did you want to take a couple of questions here about causality? Yeah. So I'll ask one, and then Seung-Min had one. You can unmute and ask the question. But in the meanwhile, so out of curiosity, I mean, I'm sure I can just read the paper, but for some of those predictions that you made, like the counterfactuals, you can actually sort of measure accuracy, right? Like the age one, you could look in a longitudinal data set and sort of see how well you made those predictions, right? So I'm wondering if you looked at that sort of thing or not yet. I mean, it feels like an actual quantifiable one.
Yeah, not yet. Okay. Actually, for this one, so the UK Biobank does only very recently have a temporal aspect, a longitudinal aspect. They rescanned people. I don't have at the moment a data set which is large enough and has longitudinal data. But yes, I mean, it would be great. I mean, and we should. I mean, they look plausible here, but are they doing the right thing?
For sure. Yeah. I just imagine you could. I mean, there's ADNI and so on. You could try to, of course, data set shift and all that, but. Yeah. Okay, Seung-Min, do you want to ask?
Yeah, sure. I think my question is along the line with Adrian's question. I just wondered that this deep structural causal model is a cross-sectional model. So I wondered that do you think that the longitudinal model would improve the results, or do you think that the DSEM takes into account the subject-wise variability kind of good enough?
I would hope so. I mean, I think actually it should be a good use case for structural causal models to bring in a longitudinal aspect, because there's a very clear direction in terms of causality. But yeah, we haven't done it. That would be future work.
Mm-hmm.
Okay. Raz, oh, sorry.
It's okay. I was just saying thank you very much.
Okay. Raz, do you want to ask?
Yeah. In the causal diagram, Ben, do you train each model separately? So I suppose there's a model for each edge there. Do you train them separately, or do you train them jointly?
So this is trained jointly. So you have the mechanisms on each of them. I guess you can think of it that so certain ones you can train independently, right? If they don't have parents, then you can train them first if you want. But they at the end it's trained jointly.
And in that case, if it's trained jointly, how do you deal with the incomplete pairs? Like, for example, if you have an image for a person but maybe not one of the variables.
Yeah, excellent question. We cannot. So the current model is assuming that everything is fully observed for every single data point.
Yeah. Okay.
It's a great one, and this would be an extension we want to look into. So this is called missingness in the problem of causality. How do you deal with samples where you don't have all full observations?
Yeah. Thank you.
So yeah, clearly a limitation of the current model.
Yep. Cool. I think we can move on. Yep. Okay.
So the uncertainty part. So I had put it at the first in the title, and then I realized it's only coming at the end. It's also recent work. So this is done by Miguel. So probably it won't take too long. But it's a very neat, simple idea, I think, and I sometimes like simple ones, really. So this is the idea is to bring uncertainty in the image segmentation, which really we're not the first. Many people looking into this. And this is summarizing what we did. So a standard neural network, if you think about your pixels here, let's say just 2 pixels, you predict, so you come down to the logits. The logits will be transformed into probabilities or labels or whatever. Then these logits are independent, but they are influenced by several pixels. We got around this by using things of like random fields in the past to get some structural consistency into our predictions, right? So we want to put some smoothness or some other structural priors onto our predictions. And what we did, we put basically dependence between the logits. And how do you do this? So the simple idea of this is it's a CNN that doesn't predict a segmentation label map, but it predicts a multivariate Gaussian distribution with a low rank covariance matrix for the entire grid. And that's the idea. So it's a really simple one to add onto any segmentation architecture. So it's basically just a layer that you change, the output layer. We use low rank because obviously you can't have that for like a full rank volumetric segmentation. But interestingly, that works reasonably well and gives you multiple plausible segmentation maps with a single forward pass. So you run your image only once through the CNN, and then you have your multivariate Gaussian from which you can sample, and you can generate as many segmentation maps as you want very cheaply afterwards. So that's the gist of the method. It's really simple, and we showed that it makes sense for so this is a toy example to motivate. So let's say you have a case of just 3 pixels or 3 parts of an image. Sorry, not 3 pixels, but 3 parts of an image. The first part is always on. The lowest part is always off, and in the middle you are either on or off, but completely, right? So that's a type of structure that is very difficult to get right with standard CNNs that would do pixel-wise predictions. Now, if you use our model with a diagonal covariance matrix that does capture the mean correctly, so you get this uncertainty in the middle, But if you sample from it, you still get these non-spatially consistent samples. And if you use the low rank on the Gaussian, you can basically capture that, and then you can afterwards sample, and you only get the meaningful samples. So that's the method and the motivation. And then we used it for several applications. So one of them is BRADS. And this is just showing that, so here's an input image. This is, let's say, the ground truth from the BRADS data set. Let's say this is what a deterministic, our baseline in this case. So this is a deep medic produced for that image, maybe not the best prediction or best example here. That's the prediction that comes out from what we call the stochastic segmentation network. So also not great, but it captures the uncertainty. And interestingly, now if you generate samples, those samples are first of all spatially consistent. And interestingly, you will often find that within the samples, there is maybe an example that's much closer to the ground truth than if you just go for the prediction. Obviously, in the deterministic one, you don't have the choice, right? You just have to live with the prediction you get. And there are a couple of more examples where you basically see a little bit the variation you get on the samples that you produce. So that's basically the visual results. The interesting thing is, because you basically end up with this low rank covariance matrix, you can do per class manipulation. So one application we envision with this type of approach is that, yes, you get a prediction, and the prediction is not much better or worse than a deterministic model. It's actually, we show with the dice course, it's quite similar. But you have a way to actually now interact with your prediction, and you don't need new forward passes, right? So it's done after the forward pass. For instance, you can choose a specific class. Let's say this active cell class, the red one, you can manipulate. So you could imagine you have a slider where you say, I want more or less of it. And you see how spatially consistent the changes are that you get on the label map. So everything else stays fixed. You just basically manipulate this one class. And then in a system where you could have like a human operator, they could like manipulate it until they get a segmentation they like. So you could, for instance, think that this is quite useful in radiotherapy or something, where you don't just want to live with the one prediction that you get out. So as I said, so we used Deep Medic here as the backend and put this on top of Deep Medic. The predictive performance is very similar in terms of dice scores on these different classes. And then we have these measures for measuring sample diversity. Obviously, in the deterministic, you don't have any diversity. So here lower is better. And this is the again showing the variation that you get in your samples. We also compared, so there are some other baselines. So there people might know the probabilistic UNet, and there's also the PHI-Seg presented last year at MICAI that work also on the same idea of producing multiple plausible predictions, but they use it in a much more complex architecture where they have a model on the representation. So I think one of them is using a multivariate Gaussian on the low-level representation, which is much more costly and more complex to train, but also the sampling process requires you forward passes for every sample. So we use this on lung nodules as well, where we then compared with probabilistic UNet and PHI-Seg. So the one take-home message from this one, which I was excited about, is this lower one, where, so you might know that in this LIDC data set, you have 4 experts per image, so 4 annotations per image. And if you train on all of them, so you just say, okay, I take all the data I get and train my network, our approach, the proposed with the low rank multivariate Gaussian, was the only one that really benefited from having multiple annotations per image. So you got the overall dice score. Interestingly, although this is still compared to one of the experts, you got better dice scores than using any of the other approaches. I'm just hearing my kids are coming back, so it's excellent timing because that's my limiting factor when the kids are back in house. So thanks a lot. That's all I have, but I'm happy to answer more questions.
Thank you. So well, first of all, yeah, thank you for the excellent talk. And there was another question that just came up, and I think it was just on the last slide, and it had to do a lot with sort of. So, Jaron, do you wanna ask it, or do you want me to?
Yeah, sure. Thank you.
Yep.
Sorry, this might be a silly question that might get lost somewhere. So I was just wondering, what is the intuition behind you want, how you want to evaluate on like multiple possible predictions? So if like how to select like the best prediction among a list of candidate prediction maps, as like if we're selecting based on like some dice score or like other overlapping metric. So why can't we just simply produce one prediction map with the best possible score?
Yeah, I mean, at test time, you don't have the dice, right? So at test time, there's no way you can do that. You don't know which of your samples would be the best. So you have to live with some sort of taking the mean or taking the expectation or whatever. Yeah, so I think this is why which is why I mentioned this idea of having a human in the loop scenario. I think a method that can produce multiple plausible hypotheses is only useful if you have someone who afterwards makes use of them, right? And it might be an expert who looks at it, or some other way of selecting which of those samples to use.
Oh yeah, and like, do we have like can we develop something like a predefined probability for like each of the prediction map? For example, like we have one prediction with like 80%, possible, like it might be a good ground truth, and have another prediction, let's say 70%, it might be a good prediction. Like, can we like further develop something like that, which might be potentially useful?
Yeah, I think what you actually want is for your samples to be diverse. That's what we try to measure with the sample diversity, which, I mean, there's little use in a method that produces 10 different predictions and they're all very similar, right? So I guess if I understand your question correctly, you kind of want really alternatives, right? So you say, oh, this is a sample, or maybe a maximally distant but still plausible sample is this one. Which one do you prefer? So I think that is actually quite important if you think about methods that provide multiple hypotheses. There's little use of providing multiple hypotheses if they're all very similar.
Okay, got it. Thank you.
I had just a very, maybe this is like one of these questions that you obviously answer in the paper, but how do you sort of balance the sort of complex, like the rank with like the complexity of the segmentation that you might want? So I assume at some point, you know, there's a limit to what the low rank can do in terms of being able to represent complex shapes.
It's a hyperparameter.
Yeah. Okay. So you just sort of tuned it.
In this first one, I think we even naively tried some. I think the low rank. So here, I think most of these results are all with a rank of 10, and I don't think we saw much difference. But to be honest, I don't think we have explored that.
Okay. No, that's fine. I guess the reason I'm actually asking, I mean, it makes sense, but you know, there's all the other models, as you said, like FISEG and whatever, they depend on these low dimensional embeddings that like deep embeddings, and they also have to deal with this. And I know that from people trying this, that, you know, that's a struggle. And so I was just wondering if you had, like, is there some advantage to this low rank? In some ways, it's somehow more clear what it's doing because we're used to low rank matrices. We know what they mean, right?
Maybe.
And so I was wondering if that affects your ability to choose the hyperparameter.
So, I mean, I think what we conclude, so these other methods, probabilistic unit and FISEG, are more complex, but they can model more. So they also can model more interesting distributions than we can. The question is whether you need them for segmentation. Maybe for others. I mean, you can think about segmentation as just one application for these things. You could do other things. But other than that, I don't have a good answer at this stage.
Okay, no, that's okay.
We are making the code available in the next couple of days, I think. So if people are interested.
Right. Okay, great. Any other questions, or should we let Ben go? Okay. Well, thank you again. This was fantastic.
Thank you.
And I'm going to follow up with some emails the same as you did about recording and all of that.
Yeah. It was a pleasure.
Thank you again.
Thanks everyone for joining. Thank you.